In the world of biology, invertebrates are a fascinating group of animals, and one of the most diverse and abundant among them are insects. Today, we will dive into some external morphology remarkable features, including the three thoracic segments, different types of antenna, mouth part orientation, the eyes, and various types of legs. The insects exhibit distinct three body parts and segments: the head, the thorax, and abdomen. The thorax comprises three segments: the prothorax, which can be found on the anterior segment, the mesothorax on the middle segment, and the metathorax on the posterior segment. Antennae serve different sensory functions for different insects. In general, the antenna might be used to detect odors and taste, wind speed and direction, heat and moisture, and even touch. A few insects have auditory organs on their antenna, so they're involved in hearing. Mosquitoes detect sounds with their antenna, and many flies use their gauge airspeed while they are in flight. In some insects, the antenna may even serve a non-sensory function such as grasping prey. The mouth parts of insects are structures surrounding the mouth that are involved in the mechanics of feeding and processing and manipulating the food so that it can be ingested. The orientation of insect mouth parts are oriented in three ways. First one is the hypognathus. The long axis of the head is vertical. The mouth part point downwards. Example is grasshopper and cockroach. The second one is the prognathus. The long axis of the head is horizontal and in line with the long axis of the insect's body. The mouth parts are directed forward. The last one is the opistognathus. The head is reflexed ventrally so that the mouth part are directed backwards between the coxa of the front legs. One example is the red cotton bug. Insects often have striking eyes. Compound eyes provide a mosaic of images, allowing for excellent peripheral vision. But there's also the simple eye or ocellus, found in some insects, which detects changes in light intensity. Insects' legs serve a multitude of functions. For some insects, the legs are adapted for walking, running, jumping, grasping, digging, or swimming, depending on their needs. Furthermore, the intricate sensory hairs and receptors on insects' legs, they play a crucial role in helping them navigate their environments, detect chemical cues, and respond to their surroundings with remarkable precision.
Arthropods play vital roles in pollination, decomposition, and are integral to the food webs. Understanding their diversity and functions is crucial for preserving our environment, reducing pesticide use, and even controlling diseases. In this laboratory exercise, we equipped ourselves with the tools and knowledge needed to explore the world of arthropods. By using baiting techniques, general hand collection, sweep net, and specialized tools like killing jars, we were able to gather a diverse range of specimens. We explored their morphology, gaining insights into their roles in the ecosystem. In this slide, we can see the materials used for this laboratory exercise, which includes 1/4 bond paper and the bait, which is a mix of peanut butter and tuna, the alcohol spray, the ziplocs, the nets, and the mason jar, which served as the killing jar that contained the corrugated board, cotton balls, and the chloroform as the charge, and as well as the test tube that contained ethyl alcohol for the general hand collection. Now, proceeding to the collection proper, we first started with the baiting technique. So we were asked to use 10 bond papers to put the bait on, and our group was assigned to put the following bond papers around the science building area. The destination of the following bond papers were selected at random. 5 of them were placed outside. So the places that were chosen was near the garbage area, in a quiet place where most students do not pass by, and also the area where plants. Whereas the 5 remaining bond papers were placed in the first floor and third floor, in the restrooms, and near the locker area.
Aidan diciamo, Aidan diciamo, Aidan diciamo, Aidan diciamo, Aidan diciamo, Aidan diciamo.
As for the sweep net, We placed them into the killing jar, which contained chloroform as the charge, and we had collected more specimens in here, including a mosquito, a fly, and a spider.
During laboratory exercise number 3, we focused on the fundamental collection of arthropods. Our primary goal was to familiarize ourselves with various techniques for capturing them. We employed methods such as hand collection, utilizing a sweeping net, and setting up tuna and peanut butter bait traps. The captured arthropods were carefully transferred into killing jars, vials, and ziplocs for preservation. After the collection process, we proceeded to mount the specimens and capture their images. To ensure comprehensive documentation, we arranged the arthropods in dorsal, ventral, frontal, and lateral view. Additionally, we made sure to record the specific location of the species captured and corresponding date. Here is an example of a specimen captured in a passive sampling, which is tuna or peanut butter baiting. This exercise enables us to understand the complexity of arthropod collection, documentation, providing valuable hands-on experience in the field of entomology.
Insect pinning and mounting is one of the methods used by entomologists to carefully observe any collected specimen in the wild. Insect mounting uses different materials. Materials such as entomological pins with paper stitch attached and entomological probe to help them move the specimen. Specimen are always moved at the end of the paper stage to help them view the different sides of the insects, posterior, anterior, dorsal, and ventral. Specimen are always moved at the end of the paper stage to help them view the different sides of the insect. Insect pinning are used in any insect that has bilateral symmetry. Symmetry that has its left side and right side up closely like to each other. These specimens are pinned at the mesothorax to help the researchers view the left side of the insect without damaging its side.
After mounting a specimen, we check its identification. We check the antenna, thorax, whether it has one or two nodes, the petiole, the abdomen for its identification using dichotomous keys. So ants are mostly wingless insects that live in groups. So what is a dichotomous key? It is used to classify organism using a list of characteristics to identify it. So the keys are designed to identify workers only. This is because workers are by far the most commonly encountered class of ants and the best known taxonomically because queens and specifically males are much less common. It is often very difficult to identify them, even in cases where the workers are common and well known. So a series of yes or no questions that we can answer by observing characteristics of living things until we have narrowed down to its identity. So the characters used in the keys are limited to those found on the outside of the body. In addition, every effort has been made to select easily observable and its characters.